Hello, everybody. Welcome to another wonderful week of virtual seminar here at Umass Boston. I'm absolutely delighted today to introduce our speaker, Dr. Amanda Bates. Dr. Bates is an associate professor and Canada Research Chair at Memorial Research University, where she comes by way of an awesome sort of route, starting back getting her PhD in 2006 at the University of Victoria. Looking at hydrothermal vent gastropods. Since then, she's gone through a number of different awesome postdocs and faculty positions at UBC, down in New Zealand, University of Otago, and other places. I met her through her really beautiful work on the Reef Life Survey, as well as then falling into a number of different groups trying to think about long-term biodiversity change. She's just a really deft thinker. I think you guys are really going to enjoy this seminar today. A phenomenal quantitative ecologist and someone who thinks deeply about meshing natural history with the sort of current environmental change that we see in the world around us in the oceans. So I am pleased to introduce Dr. Bates and take it away, Amanda.
Okay, so thank you so much, Jared. That's a super kind introduction. I think it's actually one of my favorites that I've had in a long time. So before I start, can you hear me? I just don't know. I'm basically looking at a blank screen. So, okay, I got a yes from Michael Roy, so that's awesome.
Yep.
And then can you see my slides? Yay, okay, good. So these are the two issues that I've had in the past. Okay, so today what I thought I'd do is just introduce a little bit of kind of the history of some of my thinking, and then share with you what my current lab is doing. And I'll flag at the very end some projects that individual kind of lab members are working on. And I'd love to talk about those things, and hopefully they're thought-provoking. But what I'm going to focus on today is kind of like an intellectual journey that I've had going from hydrothermal vents to basically biodiversity indicators that, you know, inform conservation and management really at a global scale. So, and Jared's right, it all started at hydrothermal vents, and I'll start there in a little bit. So right now I work at the Ocean Sciences Center, and I was really excited to come back to Canada, mostly for the wildness. But I did a kind of island tour in New Zealand and Australia, and then in England at the University of Southampton. And I have absolutely loved the wildness of Newfoundland. It's absolutely amazing, but it is seriously cold. So I'm going to talk a little bit at the end about diving, and just got to say, seriously cold water here. But I work at a marine institute. It's perched right on the edge of the Atlantic, and this area gets absolutely wave swept and wild. It's pretty amazing. But it's also about 10 minutes out of the city, and so it's a really kind of soul uplifting place to work. We basically look out of these round windows and you can see whales and icebergs, and so we try and tweet those out. Okay, so as with a lot of us, I think a lot of our research these days, and I've certainly seen this shift in my career over the last couple of decades, has really shifted to trying to understand how human beings are interacting with our world. And so we're really trying to kind of take our ecology and integrate human beings into this kind of context. And this is actually a map that came out of a working group that Jared and I were both in. But basically in this map, we condensed down across marine and terrestrial systems layers that represented climate change, human use, human population size, pollution, and invasions. And there's lots of issues with the data that goes into these layers. There's lots of kind of error and biases in them. But I think what this shows, and what I love about this map, is that human beings are touching almost every single part of our planet. And when I first started as a scientist, I did work at hydrothermal vents, and I never dreamed that we would be mining our vent systems or mining the deep ocean. And we are starting to do that now. So our reach is really the whole entire world. So with that kind of context, I think it's hard to just be an ecologist and not consider human beings in the equation. And so when I moved to St. John's, I thought, okay, what I'd like to do, and what I have is a funded program which is a Canada Research Chair, which allows me to kind of visualize a program for 10 years. I thought, wow, what do I really want to do? And what I thought I'd do is set up what I've called the Physiological Diversity Lab. And so this is basically using kind of tools and thinking from community ecology, but then also taking approaches from physiology and combining those to think about whole community responses and whether or not we can really predict change in communities using physiological data, but physiological data that is integrated across species. And so one of my main interests is really about environmental variability. And I can really tell that environmental variability is pretty sexy these days, because there's all these cool words that are coming out to describe it, like weather whiplash and marine heat waves. So we're paying attention to environmental variability, and this is increasingly important because we're changing how environments are changing. We're changing how temperature regimes are shifting, and in addition, in the ocean, we're changing kind of oxygen depletion and these other variables which are so critical to how communities respond. And so what in my lab, we kind of work on environmental variability and how these kind of different aspects of environmental variability influence from individuals to populations up to communities. And today what I'm going to do is focus on the community level. So what I've done is I've condensed down kind of what we work on into this simple, basically statement. And so I'm interested in biodiversity change. And what I'm trying to understand is how exposure to environmental variability kind of combines with sensitivity to predict biodiversity change. And are there metrics that we can extract using kind of physiology to then predict how biodiversity is going to change at a community level? And so as promised, I'm going to start at hydrothermal vents. So these are one of my favorite systems. This is a vent from the Juan de Fuca Ridge. It's about 200 kilometers off the coast of Vancouver Island on the west coast of our continent. And what we're looking at is a little chimney. There's some fluid coming out of the chimney. You can see that shimmering water. And so that shimmering is actually high temperature fluids, and that's mixing with cold ambient seawater. What's so cool about these systems is that you can basically get temperature gradients, which represent almost the full gradient of what you will see on Earth, but within meters or even centimeters in some cases. And there's really high biomass in these systems. So we're looking at these worms that probably hold the record for the most heat tolerant metazoans on the planet. They can tolerate up to about 50 degrees. And then as we move outward, we get these like chains of limpets and kind of other animals. So very high biomass, very interesting kind of systems conceptually. And so what was so interesting for me as a PhD student is I was interested in these gradients near these vents and how those gradients essentially constrain performance of animals that were near these vents. So what I've plotted on this plot is the y-axis is performance and the x-axis is thermal niche. And the reality is that as the kind of reality of climate change and climate warming in particular has, I guess, been well acknowledged and accepted in the science community, is that the physiological tools to think about and predict performance based on temperature have come back into fashion. But these types of tools were prevalent in the 50s, 60s, and then again, there was kind of a stint in the 80s, and then now. So there's quite a lot of baseline data, there's a lot of theory, and we actually know a lot about how animals respond to temperature. And the bottom line is that when something doesn't respond to temperature, it's pretty interesting, and most of the time, most biological responses are temperature dependent. And so what we can do is we can think about what kind of niche or temperature niche. We can think about temperature as a resource that's kind of accessed by animals. And there's a realized niche. So many animals will orient within a particular temperature range. So basically for that individual, you can kind of plot out the range of temperatures that a particular animal will stay within. And there's temperatures that they'll kind of avoid and temperatures both warm and cold that they'll avoid. And one prediction is that there'll be a temperature that will kind of optimize various aspects of the performance that's kind of based on energetics. So there's a maximum performance for that individual. And so behavior then orients this within this kind of range, and that's the sustained range. But then, of course, there's an upper limit for what something can withstand. And that upper limit is kind of what we think about when we think about extreme events or heat waves, because those extreme temperatures are kind of outside the range of what's comfortable for an organism. And so there's this kind of short-term heat tolerance, which is basically the buffer. So it's basically what you're experiencing versus what that kind of extreme temperature that you could withstand is. And that kind of heat tolerance or buffer is something I was interested in when I started a postdoc. And so what I wanted to do is I wanted to say, okay, well, if these hydrothermal vent systems are so variable because we've got these fluids that are kind of emerging, they're superheated, they're really hot, maybe vent animals are going to do something that's very different than a lot of other animals. So plan, and you can kind of think about this as a science plan, or you can think about this as an adventure plan. But my science plan was to compare vent animals to a whole bunch of species from around the world that were kind of had a different range of temperatures. And so basically that's what I did. And I worked with a few collaborators who funded research to go to Antarctica, as well as access hydrothermal vents using ship-based work. And so here are some hydrothermal vent critters. This is a pycnogonid sea spider, so they're absolutely gorgeous. And these guys live right outside the edges of vents in about 2 degrees. And this is this sulfide worm that I mentioned earlier, which kind of has this really high heat tolerance. It lives in fluids of about 50 degrees, right on these really hot chimneys. So a huge range of temperatures. And so to compare my kind of vent animals to fauna that were from kind of thermal regimes that were similar for this huge range of temperatures, I went to Antarctica as well as a number of kind of warm areas in New Zealand, as well as hot springs, so collected freshwater fauna, went into caves in the high alpine as well as alpine pools that were relatively cold, and kind of collected, well, it was about 80 species. And for all of these 80 species, and I think there was about 118 different populations because I collected them through time as well. For all of these species, what I was interested in is what temperature would they orient within a gradient. So basically I had a chamber that I could have at ambient pressure as well as at pressure, representing where the animals were collected in the deep sea. And I set up a temperature gradient where one end was hot and one end was cold, popped the animals in, and these are actually sea spiders, and they would orient within kind of a range of temperature within that gradient, and I'd measure that. And then I also used some small aquaria where I would ramp the temperature up and then look at when animals fell into heat coma, and then stop the experiment essentially, or stop the assay. So basically that would give me what that kind of extreme temperature tolerance or exposure was. And so what I could then do is then I could plot what the upper temperature tolerated was versus what they were selecting in that, what they were willing to expose themselves to, I think is the best way of saying this. So here we have upper temperature that's plotted on the x-axis and our upper selected temperatures. So this is what they were willing to expose themselves to if they had a choice. And the vent animals here are in red plots. And so you see a nice relationship, which you'd expect, between the upper temperature. So this is the temperature that they kind of fall into, a heat coma, versus what they were willing to expose themselves to. So now let's compare that to animals that are collected from other environments, from polar all the way to hot springs, from both freshwater and marine systems. And what I found is that these vent animals were doing something that was completely different. So if we look at, say, an upper temperature limit of 30 degrees, what we find is our hot vent animals were actually living within cooler environments. So they weren't willing to go above, say, 12 or so degrees when their upper temperature limit was much higher. And animals from other environments, which are much less variable and more predictable, were kind of willing to expose themselves to much higher temperatures. So basically, our vent animals were maintaining a much wider kind of thermal safety margin. And so that really makes sense if we think about the variability of these systems and what might kind of be evolving or kind of selecting for behavior in individuals, in animals in this system. And so this kind of work, I guess, got me really interested in kind of the thermal niche and what I can use and do with this kind of thermal niche approach. And I have to confess, some of the thinking I've done has been pretty creative and maybe even contentious. Okay, so now let's zoom out to the world. So we have our early thinking now kind of mapped out. And now let's have a think about what we can do if we have some really cool data. And I was lucky enough to get to work with Graham Egger and Rick Stewart-Smith, as well as Neville Barrett at UTAS for a series of postdocs. And what I got to work on was a project called Reef Life Survey, which I've continued on in Canada. And essentially this is a underwater kind of transect method where divers swim a 50-meter transect and basically count fish that are on 5 meters of each kind of transect, and then also count mobile invertebrates using a smaller kind of transect size. And what's remarkable about this kind of survey is that the surveys have been done all around the world, and this is actually an outdated image, so there's much more even now, including from Newfoundland. And there are now thousands of surveys that have been conducted in largely using the same methodology with people that have mostly been directly trained by Rick or Graham. And so when we're doing our counts, we're sizing the animals, our abundances are relatively within the same ballpark. And we also take images along the transect, and these data are all freely available. In addition, there's a tool now on the RLS site where you can go and basically just take a section of anywhere in the world, and it'll bring up all the species with pictures, kind of an ID guide for every single area in the world. So say you're going somewhere new and you want to know what's there, you could look and see if there's some RLS pictures from those sites, as well as all the habitat data is there for people to use and have a look at. So it's a pretty cool data set. It's pretty fun to work with. It's very well organized. And what I've done since coming to Canada is set up RLS Canada, and I will tell you that it's a lot colder here. So we've had some pretty chilly dives, but some interesting things that are emerging from this already is that Newfoundland, where we are, is pretty, I think it's pretty heavily hit compared to a lot of the rest of the world in terms of fishing impacts and some other impacts on these systems. But anyway, we've set up RLS Canada. I'm really proud of my team here because they're pretty hardcore. Okay, so let's take a jump. Let's go from individuals, which is how physiologists think. So physiologists think about individuals. Community ecologists often think about species. So can we take some concepts from physiology and apply them in a community ecology context? And so if we now think about a thermal niche and we think about now populations where each of these little gray lines is representing a population, the population might also resolve kind of on a temperature gradient where we have a maximum kind of ecological performance for a population. And now if we stretch that across the whole species, we might also find that a species has a maximum ecological performance along kind of a temperature gradient. And I have to confess that I was really influenced by working at hydrothermal vents as a young ecologist because I could see these gradients at the vents. They were unconfounded by other variables such as geographic space, and there were huge abundance gradients that were tightly related to the same temperatures I was seeing in my pressure vessels. I would see those same gradients in nature. So highly influenced. But of course, this concept that you're going to see a peak in abundance at the center of a distribution is hotly debated, and oftentimes in terrestrial systems this falls apart. So I had a brilliant PhD student who's now doing a postdoc, and Connor Waldeck basically took this on, and what he did is he used the Reef Life Survey data, and he wanted to test whether or not for how many species we would see this kind of peak in abundance distributions if we have really high quality data. And when we look back at some of the evidence from marine systems where we didn't see kind of a peak in abundance, it was oftentimes because there was some sort of biogeographic break, so that was kind of constraining the abundance of a species, or there was actually quite poor data. And so what Connor did is he looked across, I think it was over 600 species of fish, and for about 3/4 of them, he found a really nice relationship between maximum abundance and sea surface temperature. And for many of those species, that occurred kind of at the warm end. So there was this slight kind of offset in that peak, which is actually also predicted from physiological theory. So what this kind of suggests is that maybe we can use this abundance distributions to predict how communities might change. Because now we have an estimate for species within communities, maybe it'll be useful to then think about the species as having a realized niche. So here's our species. So maybe we can take this kind of peak and average it across all species in a community, which is often what we do in community ecology, is community weighted mean. And with that kind of average value, can we try and make some predictions about how that community might change under a warming scenario? And so this is called the Community Thermal Index, the CTI. And all it is the average species temperature index is what it was originally coined by Vincent de Victor for butterflies. And it's this peak in this species realized niche averaged across all species that are present. So is this useful at all? We had an honor student, Paul Day, who looked at an extreme warming event at Rottnest. So this is a coral reef in Western Australia. And we looked at the species thermal index, so this realized kind of the peak of this realized thermal niche. And we looked at how species of fish responded to this warming event. And basically what we found is that there was, all the species that we would predict based on their STI to respond positively to warming did so. Those that we would predict would respond negatively to warming also did so. So it was quite useful in that kind of context. We've also worked, Mike Burrows led this paper, and he looked at the community thermal index, and he looked at change in community thermal index in a whole bunch of fish and plankton communities across the Atlantic and Pacific. And so he looked at temperature change as well as change in this community thermal index, and what he found, so now we have just the plots of temperature change, and then the gray is actually the STI, so it's tiny little plots. But basically the take-home message is that we get this tracking of communities that closely matches what the temperature of the ocean was doing. And so these communities were all tracking warming when it occurred. They were also tracking cooling when it occurred. And when there wasn't warming, there wasn't much change in the CTI of the communities. Okay, so I guess a really key question is, well, that's great. So we actually now have a metric that seems to track kind of warming. Is it useful in any other way? And so what I'm going to do now is shift gears a little bit, because what I'm interested in is conservation. And what I was interested in is whether or not we could use the CTI to understand how protected communities were changing in ways that might be different than fish communities. And so this is work that I did quite a long time ago, but I still really love this work. And I think I just like some of the pictures of the environments that I get to show when I talk about it, because it was just so beautiful there. But this is a reef system, a shallow reef system in Tasmania. And so what we were interested in doing is working at this marine reserve. It's an incredible area of the world. If you ever get to go to Tasmania, I'd really strongly urge you to try and get to Maria Island. It's a 7-kilometer stretch of marine protected area that's also situated on an island that's also protected. So pretty unique situation. And if you look at Tasmania in a global map of kind of human pressures and impacts, it basically is relatively free of those pressures. So it kind of is a dark spot if you're looking at a hotspot map of pressures. So what's interesting about this area, however, is that it's an area of warming. And so this image shows the East Australia Current in '94 and then 2012. And this East Australia Current is increasingly pumping poleward and delivering kind of tropical recruits to the system. It's also a warm water system. And so this is looking out across the marine protected area. So beautiful kind of clear water and just amazing kind of rock formations. So beautiful on land as well as underneath the water. And in the MPA, there's been a return of, well, it's been a very successful MPA. So over the last several decades, there's been a return of large lobsters to the system, as well, this is a blue-throated wrasse, as well as many large kind of fishes. And so here we have Tasmania, we've got Maria Island, and I think what was particularly kind of special about this particular system is that Graham and Neville originally set up a really great control reference system for monitoring to compare to the MPA. So I think as we're starting to set up a lot more MPAs, we need to really keep in mind that it's not just monitoring in the MPA that's critical, it's monitoring in sites that can be a really nice reference and a well-matched reference for our MPA sites. And so there's 6 locations within the MPA that were monitored and 6 sites that were fished outside the MPA. Okay, so CTI. What I've shown here is the green is the marine protected area and the blue is the fish site. We've got time, and so this was over a couple of decades. We've got our community thermal index on our y-axis, and then we've got body size, so the maximum species length on the y-axis here through time. So what we can see is that in terms of the community thermal index, the protected area didn't show as much change through time, and we can see that using our kind of CTI. And so these are sites that are really, really close together. Outside of the marine protected area, we actually got a lot more warm water species that were coming in. And when we have a look at the body size of the animals in the fished area, which are in the blue, it was pretty constant, so there wasn't much change in the body size of animals. But the body size of the animals in the reserve increased through time. And so there was actually return of these large-bodied species to the reserve, and those large-bodied species in Tasmania tend to come from cooler waters. So this is one clue for what's driving this change or this buffering kind of effect in CTI, where our marine reserves didn't change as much, is that it was the large body species that were kind of buffering this. But there was also something else that happened. And this is an urchin, Centra stephanus, and this little guy comes in and grazes, well, it's moving and extending its range. It's now all around Tasmania, is coming up on the west side. And it creates barrens, and those barrens are wonderful habitat for species that come from warmer waters. They're used to barrens, and they're used to these kind of rocky areas. And so those warmer affinity species, there were 5 warmer affinity species that kind of came into the fished areas, and those warmer affinity species, of course, increased the CTI. So there was a combination of kind of recovery as well as the fished areas where you're removing the top predators. Those top predators weren't preventing the urchins from kind of coming into the system. And then that kind of trickled down to those urchins creating habitat that was essentially other species were coming in and colonizing. So the CTI was really useful for that. It kind of picked up this change in the community that was related to kind of mechanism and made sense. And so we thought, well, is there anything we can use then to try and think about this in a predictive way? Can we take our CTI and compare that to the temperature of the surrounding environment and try and understand whether or not our communities might respond positively or negatively to warming? And so what I've done here is I plotted again thermal niche, and I've plotted abundance to represent this abundance distribution for species. And what we might expect is that if species are kind of randomly selected in communities, and that temperature is an important driver in kind of selecting for which species are present, that we might get a close match between environmental temperature and the kind of peak of ecological performance of the species that are there. So on average, that would mean that a species from a particular community had a peak of ecological performance that matched the environmental temperature of the region. So what I've done here is I've drawn environmental temperature in red and the average, I don't actually know if this is actually the average of these curves, but the approximate average for these curves, and you get a good match between environmental temperature and these curves. And so that would look something like this, where we've got temperature on the y-axis, and we have these dots as just being imaginary dots for communities that are distributed kind of around the world. And what you would expect is a nice tight relationship between environmental temperature and the community thermal index. So on average, species within that community are well matched to the temperature of their environment. Okay, now what if we don't see that? So I'm just going to go through that before I show you the data. So if we have, now we again have our thermal index and we've got the same kind of species curves, we might have all of the species in a community that are basically all of the species in a community are found at much cooler temperatures than the environmental temperature that they're at. So in this case, the average species in a community is from a much cooler environment. And in this case then, the CTI is going to fall far below the environmental temperature for that particular location. And so you're going to have this basically deficit where environmental temperature is much warmer than probably what the species that are there prefer. The reverse can be true. So you can have an area where maybe all the species that are there within a community, for whatever reason, are actually representing areas that are much warmer than the environmental temperature. So that could be like a more subpolar site, for instance, where the species are actually representing much warmer areas. Okay, so that kind of gives a hint then to what we actually saw. And what we actually found, so the dotted line here is the one-to-one between our environmental temperature and our CTI in our community. And what we found is that in many tropical habitats or communities, that both the fish and invertebrates actually, most of the species that were in a community were found at much cooler areas. So their kind of peak in their abundance was from a cooler area. And conversely, as we move towards temperate systems, the opposite was true. In temperate systems, we have species that are actually from much warmer areas. So they're actually potentially going to benefit with warming because their maximum abundances are much higher in areas that are warmer. So there's this kind of large geographic pattern. So that was interesting. And yeah, we're still thinking about this. So the take home then is that, well, John Bruno actually took this and turned it into a lovely paper that's been cited many times. When he first told me about that, I was like, seriously, we're gonna write a paper about MPAs and basically highlight that climate change is gonna impact MPAs. And that's exactly what we did. I thought it was pretty obvious, but turns out it's actually not to a lot of people. And literally this paper went viral in the media. And what we used in this paper is our CTI. So we used our CTI for a lot of the marine's protected areas and showed that many of our world's marine protected areas have this kind of negative thermal bias. So a lot of the species that are in these areas are from areas that are much cooler. So they're already probably warmer than what would be ideal. And then in addition, we saw that a lot of these marine protected areas are just going to be hammered by warming as well as deoxygenation and acidification. Okay, so the kind of journey in our thinking then kind of led to this concept of climate resilience. So we have marine protected areas that are essentially being hammered by environmental parameters. And is there a way that we can think about the physiology of the species that are there to kind of make some predictions about what we might expect? Because one of the big challenges that we're facing is that if we're protecting areas of our coastline and we're preventing kind of fishing or other livelihoods, there's obviously some kind of conflict that can evolve around that. And if those areas are then essentially wiped out in extreme kind of weather events, then all of that energy and the setup of those marine protected areas has essentially been lost, and livelihoods are negatively affected. And at the end of the day, I think we'd all argue that what we want to look out for is human well-being. And so what I've done here is I've just shown a plot from a paper that we published, and we coined a term, which this is actually a well-known concept, but because I didn't come from a conservation background, I was actually a little surprised when I first thought about this. So I thought, well, if it surprised me, then I'm going to put it into a manuscript. So what I've done here is I've shown in green a little schematic that's areas that are protected, and then in gray it's unprotected areas. And on the left-hand side are basically some coral communities that are dominated by parietes. So let's imagine now that we protect these communities and we get a regrowth in our protected areas of these foliose kind of corals that are relatively sensitive to things like anchors and gears. And so we get a regrowth of these coral species. These coral species, many of the branching ones, also have a much lower STI, or this kind of thermal index, than the rest of the species, than parietes. So parietes are much more heat tolerant. And so this has actually happened in a number of marine reserves where the reserves have recovered over a decade or so. And then there's a big heat event, which I've shown in orange. And all of the species that are recovered are basically lost. So they're bleached and they die. And this is especially, there's one marine protected area in Kenya that is showing this. And it's also in an area that's very exposed to heat waves. So if the managers of that reserve had just used a simple tool like our community thermal index and thought a little bit about the exposure of this community to heat waves, this actually would be quite predictable. And we coined this the protection paradox, and it turns out that this is being picked up in many different systems, including terrestrial systems, because this kind of effect is being observed, I guess, around the world. And so the utility of this thermal index is basically, it's now a biodiversity indicator, and we're feeding it into initiatives, including the SDGs, as well as some of the IG targets. So it's a BIP indicator, and there's a huge amount of information that kind of supports this index, and a lot of that I've chatted about today. But I think it's quite useful, especially in systems where we've got large geographic ranges, we have continuous recruitment and connectivity in our populations of species that are there. So it's quite useful, I hope. And it's been really fun to think about, and also to think about how kind of creative thinking can then be turned into something that's kind of useful for conservation. So I'd like to encourage everyone to be, don't be afraid to be creative. Even if people think it's a really terrible idea to begin with, after a while, they often don't. And especially if there's a really solid reason to use something and you basically practice explaining things until people understand. Okay. So what I'm going to do now is, that was just like kind of a little trajectory of how we went from really pure kind of ecophysiology at the individual level up to a biodiversity indicator. And what I'd like to do now is just share a little bit about what I'm doing in my lab. And a lot of what I'm doing in my lab really overlaps with what Jared does. So I'm sure that we're just facing a huge amount of challenge to basically access information about environmental condition. We're challenged to even get adequate data about fine-scale environmental variables and human pressures that are relevant to communities and individual populations. So this is a challenge. When we combine that data into kind of global observation networks, I feel like sometimes we split our efforts. So we're trying really hard to come together as a global community, but all of this takes communication and time and commitment. This can be hard to get people to buy into when we're already so committed with other things. And then as we kind of take our ecological data, ideally what we want to do is kind of match those up at scales that are going to match with other variables that we need to kind of understand the context of what we've collected, and that the scales at which we're collecting this information is going to be well matched. So now we're getting into kind of an interdisciplinary framework. And then we need to be kind of skilled at using statistical approaches to combine these kind of multiple data sets and then transform them into decision making. So now we're going into kind of a transdisciplinary framework. And all of this takes an extraordinary amount of communication and commitment and time. But doing so and then working together as a team means we're going to lead to improved kind of environmental outcomes, at least I hope. And so a lot of my work now is really working in a transdisciplinary framework with really big groups of people, where we're doing things, for instance, like working in the Arctic. So for instance, with the Nunatsiavut government, so it's Inuit self-governing nation. And we're implementing a marine geonode, which Jared will know about. And we're doing that in Nain in the Arctic. So that's really exciting, but it also takes that vision to kind of integrate that sampling within a framework that actually is doing something completely different, because what the grant is about working with Inuit youth. And yet what we're doing with our Inuit youth is implementing a survey node. And instead of making up a new survey node, I decided to work within a project that was already underway and already had momentum, so that that data and information we can collect can then feed into our global initiatives, so that we can hopefully increase the spread and extent of our knowledge. So as mentioned, it's actually largely, I have to say, the passions of Jasmine Schuster, as well as many of my students. All of the students in my lab work on some sort of global collection effort. So they work on BioTime, which is a biodiversity time series database, or Reef Life Survey, where they're going out and collecting data and feeding that into a global initiative. And those kind of efforts then sometimes can be completely independent from their thesis, but I think it's a really rewarding experience because they increase their global kind of network. And then, I mean, let's face it, everybody loves doing something else besides their thesis when they're doing it. So this is like a really productive and fun thing to do. So we started Reef Life Survey Canada. We have our 3rd year rolling where we're monitoring sites. And we have a really kind of low diversity system, which is really easy to count. We have beautifully clear water here. It's absolutely gorgeous, so we don't get a lot of plankton. So it is really beautiful diving. And what I'm doing in my lab is I'm interested in how humans kind of tinker with physiology. And so what I'm going to do is just take you through 6 projects and just highlight those projects in less than a minute each. And then I'll close, and hopefully we can have a great chat and about some of this work. So I think when this really started kind of crystallizing was a few years ago with Murray Duncan's PhD work that he did with me. And what Murray worked on was exploitation and how that influenced the physiology of fish in marine protected areas. So this was at an individual level, and was just a lovely set of experiments and work that Murray did. And basically he worked in a marine protected area, which is in South Africa. And this is a huge marine protected area. It's like 50 kilometers, and it's very well governed. It's very old. And he compared a species of lionfish, and this lionfish has quite high site fidelity. And so what that means is that these populations in the MPA had many kind of generations to evolve, really, or I guess avoid selection by fishing. And so what Murray did is he collected animals and did experiments where he looked at aerobic scope, and that's what is plotted on this y-axis. And basically he looked at a number of different temperatures, and the blue is the protected area in this case, and the fish is in red. And what he found is that in the protected area, we have these really kind of high performance physiological kind of phenotypes or genotypes, we're not sure. And basically these individuals have a higher aerobic scope at higher temperatures. So basically they have a slow metabolism. And if you think about this, this kind of makes sense, because if you're fishing individual fish, you're removing the fish that grow the fastest, because the fish that we're removing from the ocean are the biggest individuals and populations. So probably what's happened here is that these really slow growing fish can kind of, they're basically protected in these areas. And this size selection that fishing imposes is also very likely influencing physiology through metabolism. So that was kind of the start, and what I've really launched in Canada is everyone in my lab is really working on physiology and how kind of humans and human activities influence the physiological diversity of communities. And so here we are in this crazy circular building, which is beautiful but impractical in terms of setting up a wet lab. But anyway, absolutely gorgeous site. And I have one student, Valeska. She's a year into her master's, and Valeska is interested in cod. And of course, cod is quite iconic here. And what she's looking at is how our last winter influenced the phenotype of cod in a population here that's been monitored for quite a long time. And we're using a really cool technique. I'm happy to talk about it later. It's called field metabolic rate. And what field metabolic rate, it's a new technique that's just been developed in the last 2 years by a colleague that I happen to have at the University of Southampton. And what it actually does is it allows you to calculate the energy use on a daily scale using the odolith. So we can actually get what's called the field metabolic rate, or what the energy expenditure was for our fish population before and after the winter. So we can look at how the winter basically kind of selected for specific individuals within the population, which is quite cool. And we have Brandy, and so Brandy's interested in the green crab, and she's interested in the basically what the green crab has been doing in terms of population trajectory from around the world. So she's extracted population abundance data from green crabs in their native and invasive ranges, and she's looked at either edge of the geographic range, and she's been looking at the warming and cooling trends in all of these areas. And basically some of the take home is that the green crab is actually quite sensitive to warming. And so everywhere that it's warming on the warm end of the range, the crab is decreasing. And overall, in spite of a lot of kind of theory predicting that invasive species, because they're more likely to be more physiologically robust, are going to increase with warming, we haven't seen any evidence of that looking at the green crab with about 50 years of data from around the world. So that's kind of a cool project, which is taking some of the macroecology that we've done and thinking about it now in this kind of more applied framework. And Jasmine and Jarrett, this is the project that I think you might think is pretty interesting. Jasmine has been looking at urchin barrens and kelp forests, and she's looking at how urchins influence the physiological diversity, both at the community level, but also at the population level. And so she's interested in how urchins reshape the physiological diversity of communities, which is pretty fun. And Jackson was in my lab. He did a postdoc with me, and he went to Antarctica as well as 2 sites, Arctic and then temperate, and looked at the response of invertebrates to changes in oxygen. So he was looking at what's called the peak crit. So this is this threshold when animals basically lose their capacity to regulate oxygen. And because of so many oxygen changes in kind of our global ocean, Jackson was kind of working on that project, and he did this for more than 20 species with 10 individuals for each species, and developed these kind of beautiful peak crit curves for all of these species. And now what we're doing is we're looking at that kind of in a community level, and using these peak crit curves to predict the outcome of kind of oxygen events. So that was also a fun project, and is kind of scaling up what we normally do with physiology by generating kind of single curves for individuals, but at a community level. And then Karen is working on seabirds. So she's finished her master's with me, and she's interested in traits. So she's interested in how traits are influenced by fishing and fishing gears, and whether or not we can use those traits to kind of predict the outcomes of different anthropogenic threats. And she's now gone on to do, well, she's just started her PhD, and we'll be looking at seabird energetics. So we'll be, it's the first time I've worked with like something that's warm-blooded. It's quite exciting, but we'll be looking at energetics and then trying to predict the outcome of the response of seabird populations to kind of variability and extreme weather events. Okay. So that is what I'm doing in my lab. Hopefully that will contribute to some of this kind of overall objectives to create improved environmental outcomes for the globe. And of course, there's a lot of funders and a lot of wonderful, wonderful people that I've worked with that have shaped my thinking along the way. So I'll end there. I really hope that everyone could hear that.
That was great. Thank you very much. That was really wonderful. We did hear. Wonderful. So if people would like to ask questions, we have the Q&A box open. I haven't seen anything come in during seminar, so feel free to use that right now. Ask a question there, and I will read it out. Give people a second for that. And actually, I'll start with just sort of a bunch of questions. It's a really cool talk. Thank you so much again.
Thank you, Jarrett. You're always so positive.
So, all right, let me bring my question list over here. Yeah, so I actually, okay, a lot of things. First thing, I was really curious about the vent story. And one thing that I thought was really remarkable, we were talking about that these vent species are often choosing cooler temperatures than we would predict based on, for their sort of average range relative to their maximum as compared to non-vent species, right? I thought the shift in those two curves was really interesting. But what it made me think of was maybe go back and really think about rocky intertidal ecology and think about salt marsh intertidal ecology, that many of these species are existing at these extreme environments because of competition at the end of the day, that they actually prefer more benign, or I guess in this case, cooler conditions, but they're the ones that can hack it at those more extreme conditions. Is that sort of what's going on? Is there sort of a similar story there? And kind of, if you want to jump off from that, thinking about that in the context of like Chris Harley's work, where he's showing how that sort of stress gradient is changing and how that's changing the whole zonation of the intertidal. I do think there are connections there for these communities.
Yeah, so I guess at vents versus the intertidal, like there's a productivity gradient. So the harder you go, the more productive that environment is going to be for the bacteria that are there. Mm-hmm. So if you think about that, if you think about the physiological cost of living in those environments, the harder you can take it and the more you can access that productivity, the more energetically favorable it's going to be. And I actually have a student that's just started who I didn't add to this talk, but she's going to be developing hopefully the first dynamic energy budget model for a vent animal. So, okay. So now let's go to the intertidal. So in the intertidal, you have kind of a shift, and a lot of these animals are really shutting down. So, I mean, if you think about how much time the intertidal animals, especially in the high intertidal, are actually active, it's not a huge amount of, I mean, Jared, if you think about it, like they just completely, you know, they switch on and off depending on the tides, right? And so I'm just trying to think about how to think about this. And then the conditions are quite variable between those times. So what I guess I would think about is, how do you characterize what is ideal for an intertidal animal? Do you use the conditions where the animal is like basically in kind of an anaerobic state and is shut down, like basically it's hibernating, or the equivalent, you can think of it like that, or do you use the conditions when the tide's up? Mm-hmm. So I think that competition shifts these, but when we at least looked about it with the reef fish, mm-hmm. competition is absolutely coming into play, and you really see that between the tropical and the subtropical kind of transition. So what I think's going on, And you see that at vents too, with some of the species that I looked at with the gastropods. There was this shift in their kind of peak in abundance. So I'm going to guess that like 1/3 of species are affected by competition or some sort of species interaction, and that shifts. But then there's, I'm just going to guess, like 2/3 aren't. And so you can still take the middle of their distribution and have that be an estimate of, at least at the community level, of what their, it can still be predictive at a community level, if that makes sense.
Yeah, yeah, that makes a lot of sense.
Yeah.
Actually, to follow up on that, Breck McCollum asks a really good question that I think is a natural follow-on here. So she says, I'm struggling to put my hard-fought physiological data into a macroecological framework. I love the CTI concept, but I'm wondering how to apply it to a system that experiences a wide range of temperatures on a tidal cycle, like tidal flushing swing temperatures. She's talking about subtidally, though, rather than intertidally. We get some pretty big variation just on tidal swings at 10 meters, 20 meters.
Okay, I should say, Jared, I should announce this now. I am working with an honors student, and what we're doing is collecting fine scale temperature, oxygen, salinity, and pH data, and we have a big database. And if anyone has temperature that has been recorded at less than like a frequency of 2 hours or less, we'd love to collaborate with you and have you input to our database. It's called HOTS, just so we can email people and ask if they have the HOTS for us. But I think it stands for something like high frequency oceanographic time series data. And we're getting some really amazing ocean weather kind of patterns. And I forgot to mention that. But anyway, so I'm not really sure. Like, I've thought about this quite a bit about what to do with this kind of data. And so I think what you can think about is, like, what is that? Like, think about it in terms of the kind of energy and what that animal is doing over the temperatures that it's experiencing. So for instance, at vents, animals I don't think they shut down. I think they just have a really broad tolerance. And so vents are highly variable. So probably what that means is that the animals are performing at, like, basically their physiological machinery is mismatched to the temperature that they're experiencing. So energetically, they're probably not efficient. So I think it depends on what your animals are doing. Are they tracking temperature? And then are they fine-tuning their physiology so that they're matching, and they kind of like basically are they working within the window of temperatures and conditions and then shutting down when things are suboptimal? Or are they tracking those conditions through time? And so I guess what we're trying to do is think about this as an energetic problem, and then trying to use models, energetic models, to predict what, or at least predict what then you might see going, and you can kind of get from an individual physiology to a population level. And then if you have multiple populations within a community, you can kind of go up to the community level.
Interesting.
I don't know if that answered that question. It's something that's hard. I don't know what else.
No, I think the questioner, Breck, will have a lot of things to talk to you about afterwards, including a 30-year high frequency time series.
That would be awesome. You can join our HOTS database.
Great. So let's see. Next question. This is from Ron Eder. He says, thanks, Amanda. Is it possible the difference for the hydrothermal vent organisms reflects other gradients at vents, such as hydrogen sulfide, that co-varies with temperature? I think you dug into this already, but.
Yeah, I mean, yeah, I have. So it could be sulfur, reduced sulfur, oxygen, sure. And it could be that it's exposure to these kind of other toxic chemicals for sure that affects them. But the temperatures that I measured, like in my gradients, were like, I didn't have chemically altered fluids. So it was just seawater. And it's actually pretty interesting because with the venomals, I did a bunch of Y-maze experiments and stuff with sulfur as well as oxygen, and they just don't even respond to these variables at all. And you can even hold these sulfide worms like in your basement for months with no oxygen. So I would actually love, like, no one's really working on these animals, and they're doing something very different. So it could be that it's avoiding these kind of toxic kind of fluids, but the vent animals do something very different. So when I was working with them in the chambers, what happens is that when you kind of heat them up, they fall into a heat coma. If you bring their temperature down, they wake right up. And vent animals do that more than any other of the other species that I worked with. So they have kind of a recoverable heat coma. If you fall into a heat coma at a vent, you just fall off the vent and you fall right into cold water. So it could be almost a response to that. And the other thing I noticed with vent species, and because I started with vent species and actually kind of thought of doing the large scale study after I did the cruise with the vent critters, I probably could have designed the study to capture this, but they have these really amazing escape responses. So when you heat up a vent animal, if it gets really hot, it, like a worm, an albanellid worm, for instance, will jump out of its tube and start to swim. And when I went to the Antarctic, I didn't really see that at all. So unfortunately, I went from hot to cold in my experiments. But it just completely struck me when I went to the Antarctic, like how thermally dumb the animals were. So I do think you're probably right that it's also the toxic compounds with the fluids. But what was interesting is how programmed those animals are to respond to temperature, which I think was key. And the other compounds are actually really hard for animals to detect and respond to. And probably temperature is the safe one if you're going to predict what the fluid chemistry is going to be like. Temperature is what ecologists use at vents too, largely.
That's great. Great. I think we've got 2 more questions, and then I'm actually going to put in the chat right now the link to the reception afterwards, so where we'll go after. Let's see, from Bill Hager, we have the question of how do otolith measurements give you energy values?
Okay, so I'm not a geologist, but my colleague who is a geologist, basically it's the carbonate chemistry. And so you basically take the otolith, you ground it up, and you basically look at the isotopes. And then what he did is he worked with cod, and he held the cod at different temperatures. And then that basically leads to a fractionation in terms of the isotopes in oxygen. And of course that can then, through a bunch of math, which I don't understand because I didn't generate it, but basically you can back extrapolate what the oxygen consumption was. And then from there, you can then leap to metabolism. So that paper was published in Nature Communications. The lead author was Chung. But if you just put in field metabolic rate in fish, it just comes right up. And since then, so this was a couple years ago, it's just completely taking off because, like, think about what this means. Like, you could get daily energy expenditure. Not only that, you can get temperature. So from that chemistry, you can actually get the temperature that the fish was experiencing. So your fish becomes like a little temperature logger, which is pretty cool too. So people are now using it. Like, I know Kevin McMahon's lab at Guelph is working with this in freshwater species. There's people doing it with Pacific cod. That's just what I know about. People are going back now and taking otoliths from like fisheries, like Department of Fisheries and Oceans in Canada, and for cod, for instance, and then looking at what the phenotypes were of cod historically and how those have been shifted, for instance, by fishing, which is a super cool project. So, but Clive is really open. So it's Clive Truman at Southampton University. He's a great guy, and he's super open to discussing this. And he's a geologist, and he really wants ecologists to use what he's done with his team. So he is more than happy to talk to you.
Great, thank you very much. We have a number of questions left, but I realize the time is getting on for many of you. And so in the interest of honoring your time, we'll move this over to the reception afterwards. So everyone take a break. and we'll reconvene in a more relaxed Zoom venue. I don't know if you Zoom venue.
Yeah, and I can't see anyone, Jarrett, so I'd love to talk to people individually, right?
Better for everybody. Everyone will have their cameras on. I actually have to make a pizza while we're doing it, so you know. So the link is below, or if for whatever reason you can't get to the link that I pasted into chat, it's in the email from Sarah, or you know, email me, text me, but the link is right there. So we will reconvene there in just